Thank you so much for the opportunity to be here. We're very excited to talk about Aura and the new technology. I'm a urologist by training. I've been faking an ophthalmologist for the last year, but that is fine. The eye looks like a small bladder, so we will be just fine. So I'm going to go ahead and get started. So talking a little bit about suprachoroidal injection and our technology and where we are in our clinical program is super important, but I think we need to back up and talk a little bit about Bel-sar, what is Bel-sar, and what is Aura doing to leverage then the suprachoroidal delivery. We are the first company and the first in its class to be using a viral drug conjugate. Our founder and CEO, in combination with the NIH, the same lab where the Gardasil vaccine was founded, has taken the human papillomavirus, removed the genetic material, and then added to the viral capsid a small molecule protein payload. So what's really exciting about this is when you put it together, it's highly specific and highly specific to binding cancer, and I'll show you that mechanism of action in a second. So if you have this viral drug conjugate, in our preclinical models, you've seen that there's over 138 different types of tumor cell lines where the viral drug conjugate binds very specifically, and many different types of cancer lines. And why does it do that? Cancer cells and damaged tissue cells all express HSPGs, or heparan sulfated proteoglycans. You don't see that in normal healthy tissue, but you do see it in damaged tissue and cancer lines. And so when you inject Bel-sar into that environment, it will bind very specifically to the cancer cell lines, but not any of the healthy tissue, making this a very highly focused and very specific technology. So the mechanism of action then. When you have that highly specific binding, you have Bel-sar, and you have, again, that small molecule payload. When you hit it with a cold 680 nanometer laser light, it will actually activate that small molecule to recruit reactive oxygen species and immediately cause cellular necrosis. It's pretty amazing. The cancer cells die and die very quickly, and the healthy tissue is not affected. So that's mechanism of action number one, that very specific local cancer cell killing. The other is, like I'd like to say, for lack of a better medical term, spilling of the guts. We open all of those neoantigens to the extracellular environment and allowing for the immunology, your own host immunology, to come and gobble up those cancer neoantigens and then induce a CD8 positive T cell, memory-based immunologic response. And so I encourage you to take a look at our website, look at some of the new data we've just put out on our bladder program, and you can see that we have our first pathologic samples in vivo of humans showing some of this potential for an abscopal effect, which is really exciting. We haven't obviously been able to demonstrate it in the eye for biopsy-based reasons, but in the bladder we can show it. And so it's really exciting. We have this dual mechanism of action, and down the line, will there be this immunologic response to both local cancer cells as well as micrometastasis? Time will tell. So going into the suprachoroidal delivery, some of you may know a little bit about Aura's history. The phase 1 first program was done with intravitreal injections, and the efficacy was mixed. So the company went backwards and said, what else can we do? And then they found, in combination with Clearside Biomedical, the suprachoroidal delivery opportunity. So here you can see some of our early models in the orthotopic rabbit, both if you have the tumor and just saline injected versus tumor plus Bel-sar, and activated Bel-sar, the tumor is completely gone. And then you can see that there is presence of Bel-sar in that suprachoroidal space. And on OCT, you can see that there's nice distribution of the drug. And so very similar to other images we've seen here, the suprachoroidal delivery does do a very nice job getting the drug circumferentially to the eye. And so the thought was that we can better bathe or coat those tumors in the choroid with the suprachoroidal injection versus the intravitreal injection. And that's what led us to the phase 2 program using SC technology. So let's take a look at the phase 2 program. The phase 2 program had multiple cohorts, but ultimately if you break it down, one cohort was what we consider to be a subtherapeutic cohort. The other one was 80 micrograms, or a therapeutic cohort. And this is very similar to what is being done in our phase 3 program. And so after the completion of the phase 2 program, those 10 patients who received a therapeutic dose had 80% complete tumor control at 12 months. Versus in the subtherapeutic dose, only 20%. So really exciting data showing no more tumor growth, very similar to what you would see with plaque brachytherapy, and leading us to the next step as an organization. But what about visual loss? Because that's really the secret sauce here. We know plaque brachytherapy is very good at controlling tumor growth in choroidal or uveal melanoma, but it can cause vision loss at a high percentage. So here we saw 90% visual preservation at 12 months. And so the goal here with Bel-sar in uveal melanoma is to control the tumors locally, but also to prevent vision loss. And it's very safe. You can see there are some small amounts of grade 1 AEs, but as compared to the intravitreal injection, there are far, far less inflammatory-based AEs. Overall, most were trace. They were very short-lived in terms of the adverse events, with minimal or no treatment, and they were, no patients had any serious adverse events. So the company has now embarked on a phase 3 global, multi-centered program for the treatment of choroidal melanoma called the COMPASS program. We're looking at small tumors of 0.5 to 2.5 millimeters in thickness and no larger than 10 millimeters in largest basal diameter. So I encourage you, if you have patients with these small nevi or potentially CMs, where you've seen them in your office, and you'd say, hey, vision preservation may be an option for you if you can be enrolled in this trial. Take a look, scan this, send this to one of our sites. We're actively enrolling now, and we truly hope to get this patient population a new treatment option and preserve vision. Thank you so much for the opportunity, and we're very glad to be here.